Okay, so today we're going to start with station number 3, excretory system. So you should have filled out this part by using the slideshow in the Nearpod, and now we're going to begin with the blood filtering activity. So you can see we have a baggie here with blood components, and it asks you to fill out the number of each of them present. So I'm going to count out each bead, fill it in, and show you when I'm done. Okay, so here are the components of the blood. So red blood cells 9, white blood cells 6, proteins 5, amino acids 10, glucose 11, salt 13, and urea 14. For this next step, it says add the contents of the bag labeled blood components to a cup labeled blood in the renal artery entering the kidneys. Blood also contains water, so add enough water to fill the cup containing the beads. I'm just going to cover the beads with enough water because we don't have very big beads. And then I'm going to pour the blood component into the artery cup and add water. And then we're going to lay a screen over the bowl and then pour the blood over the screen. Okay, so here I have my bag of the blood components. I'm going to put that in the cup that is labeled. This says blood in the renal artery. And here's my cup of water that I'm going to use. And get all the blood out. Put the water in. Okay, so now we can see the blood. Okay, so then we're going to take this. We have the screen. So this is going to be kind of like what your kidneys do. They screen or filter things out. So I'm going to put this down with the screen here. Okay, and it says lay the screen over the bowl. Pour the blood from the renal artery over the top of the screen to form a single layer. Pour the material that stays on top of the filter into the renal vein cup. So I'm going to pour the blood from the renal artery. As you can see, after I have poured it over, we have all the little beads down here and the big beads on top. So the next part says to pour the material that stayed on top of the filter into the renal vein cup. So that's this cup. So I'm going to pour these back in here. Number 9 says, most but not all of the substances leave the blood. What 3 components are part of the blood? So the only thing that made it back into the vein were these, the red, the green, and the white large beads. So the red, the green, and the white are red blood cells, white blood cells, and proteins. 10. Some essential molecules such as glucose and amino acids are kept by being reabsorbed. These molecules should be completely returned to the blood and should not end up in the urine. The kidneys use energy to transport these molecules back into the blood. What 2 substances in the filtrated are essential and needed? Well, that is the glucose and the amino acids. Step 11 says model complete reabsorption. Use transport protein spoons to return the glucose and amino acids from the bowl to the renal cup. So we said glucose and amino acids. So the glucose is the little blue ones and the amino acids are little green ones. So I'm going to take a spoon and I'm going to go in to here and remove the green ones and the blue ones from here and put them back into the blood. Number 12, other molecules such as water and salt are balanced by being selectively reabsorbed to maintain proper salt and water balance in the body. The reabsorption is regulated so that they are returned to the blood if needed, but excreted into the urine if present in excessive amounts. Specific transport proteins in the nephron use energy to move these molecules from one nephron into the capillaries of the surrounding nephron. So what two things? So that was salt and water. Number 13 says model selective reabsorption and return 5 salt molecules to the renal vein. So now I'm going to take 5 salt molecules, so the white tiny beads, and put them back into the blood. The next step says transfer enough water of the nephron to fill it up halfway. So we're going to give half of our water back to the renal vein. And what didn't get transported, so what didn't get transported were the yellow ones. So the yellow ones, if you look back, the yellow ones were urea. Okay, so this is called urine. So the blood in the renal vein cup contains the cleaned blood. So this is all of our clean blood. So after all that dirty blood went through the kidneys, okay, we filtered out all of the good things, and now we have what the body is going to need. So after reabsorption has occurred, what substances are present in the clean blood? So we have the large green, red, and white ones. We have the green, blue, and white small beads. Okay, so I marked out, we have our red blood cells, our white blood cells, proteins, amino acids, glucose, and salt. So those were the things that are in our clean blood. It said what molecules in the blood or the beads were able to be filtered through the kidneys. Should all the molecules move through the kidneys? Which ones should not? So when we did the filter, we noticed that the big ones didn't get through. So the red blood cells, the white blood cells, and the proteins did not get through. Okay, and the ones that did get through, that were filtered out, were the yellow little ones, the white little ones, the green, the blue little ones. So all those little tiny beads that represented those smaller things like amino acids, glucose, salt, and urea. Okay, and for number 2, does the process of filtration alone completely separate the waste from the essential materials? Support your answer with observations of what is presented in the nephron cup. So remember, we had to add some of the little beads into the final cup because this is what was caught by the filter. But I had to move a little bit of salt over, and I had to move all of the blue and the green ones over, which were the glucose and the amino acids, because those need to be reabsorbed by the body, and I had to use the spoon to do it. So this was not automatic. This has to be something that's done by a specific process.